So, uh, and that's the acid-fast stain that we're talking about. So the next particular attribute of bacteria that's pretty important to understand is that of motility. We've talked about this before as well. Some bacterium have the ability to swim, quote unquote. And if they do this, then we refer to that as being motile, which means that the bacterium is able to get from point A to point B using particular attributes or motile structures that allow them to do that. And the key structure for this is typically the flagella, although there are some organisms that do, like I said before, a tumbling motility, or they might move through a twisting type of motility. So those types of motilities are also possible. Most spiral-shaped bacteria and about 50% of bacillus-shaped bacteria are going to be motile. So that Treponema palladium, where we saw the twisting natured cell of the bacterium, those would be the ones that you would actually be able to see move if you were viewing them under the microscope. And most coccus bacteria are actually going to be a non-motile type of bacteria. We can observe motility when we place bacteria inside of a semi-solid medium, such as like gelatin or some sort of nutrient agar media that is kind of, just a semi-solid, so half solid, half liquid. Or you can also use something that's called the hanging drop technique, where you're essentially placing a drop of your bacteria in a sample of water, and you can view them underneath the microscope. This is the semi-solid agar method. Here is a test tube that contains media inside of it. And inside of this media, what you do is you take what's called an inoculation stab, which is basically a long metal needle, and you have some bacteria sample on the needle, and then you inject that into the media. And then after injecting, you let the bacteria grow, and you record your results. If the bacteria grow inside of the stab that was generated with the needle, then that's referred to as non-motile. But if you see after 48 hours that your bacteria have spread throughout the media, then we refer to that as motile. So this is a semi-solid agar method. For hanging drop technique, you simply have a depression slide, which is a slide that has a little dip inside of it, and then you add your bacteria inside of a water sample inside of the depression slide, and then you cover the slide with a cover slip. And to do this, you actually take the slide and place it on 2 things of petroleum jelly. And once you have this set up, you view it in the microscope, and then you can look for movement in the bacterial cells. For colony morphology, whenever we observe colonies of bacteria, we typically grow them growing on what's called a nutrient agar plate, and all the bacteria that we grow on plates typically reflect a certain different type of colony morphology. And this allows us to determine whether or not we have more than one species of bacteria growing on a petri dish plate. So in one bacterial colony, you can have millions upon millions of bacterial organisms. The colony morphology, as I said, is going to be different in comparison to all the species that are out there. So things that you evaluate are going to be things like size, color, the shape of the colony. You'll look at the elevation, so how high or how low does the bacterial colony raise itself above the agar. And then you can look at things called the margins or the edges of the colony, which will dictate a specific kind of pattern. So you might see some fluffy edges or giant lobes on the edge of a colony, which we refer to as lobate. You could also see some serrated edges that look kind of like the edges of a saw that you would cut a tree branch with, and we would call that kind of a rhizoid kind of look, or you could just have irregular margins. The most common is going to be that of smooth margins, where it's just completely smooth all the way around, and another term for that is called entire. Colony morphology also includes results of enzymatic activity on various types of media, and what that means is we add certain pH indicators to a media, and depending on how the bacteria react to the enzymes inside the agar or the substrates inside the agar, such as like sugar substrate, that'll determine basically what type of color is being present. So if you wanted to see if a bacteria digested a specific sugar like glucose, you can add a pH indicator called phenol red to the media. And then if the bacteria digests the glucose through the process of fermentation, then the bacteria will release a lot of acids in the media, and the acids would cause the pH indicator to turn from a red color to a yellow color, indicative of an acid pH being present in the media. So that can be typical in terms of exploring enzymatic activity using different types of media. So the colony types will change. Colony morphology is pretty important for us to be able to identify bacteria, which is why when we want to figure out what type of bacteria somebody has been infected with, we have to culture them on nutrient agar plates as well as in nutrient agar test tubes and differential medias, which are medias used to help us distinguish between different bacterial species. So the way that colonies grow is that the size of the colonies are dictated by the organization or the organism's generation time. And by generation time, this is basically the time taken for one cell to become 2 cells, or the time taken for 2 cells to become 4 cells. Bacteria grow exponentially, which means that they're going to go from 1 cell to a multiple of cells. So 1 cell to 2 cells, 2 cells to 4 cells, 4 cells to 8 cells, 8 cells to 16 cells, etc., etc. There is a generation time of bacterial cells is considered to take place every 30 minutes. So every 30 minutes you have 1 bacterial cell that's going to change into 2 bacterial cells. And so if you look at this diagram here, you can see you have a single bacterial cell at time 0. That's going to be 1 cell. And then after 4 hours, we end up having 256 cells. And the reason for that is because you have this exponential growth rate of the bacterial cells every 30 minutes. So if you do the math on that, then you end up with a number of cells is equal to 256. And then after another 4 hours, you put you at time 8 hours, and then that'll give you 65,000 cells. And then after 12 hours, that's another 4 hours, you end up with 17 million cells being present. So that's how colonies grow. There are certain atmospheric requirements that bacteria need in order to survive. Bacteria can be classified on whether or not they're able to use oxygen in order to undergo cell respiration, or if they're going to use, or if they're going to be able to thrive without it. But oxygen and carbon dioxide are still 2 important factors that are used to help us classify bacteria. So bacterial organisms that rely on oxygen are going to include obligate aerobes, which means these are bacteria that have to have oxygen to breathe or cell respirate. Microaerophilic aerobes, they need just a little bit of oxygen present. Facultative anaerobes, this means that these organisms are typically fine if oxygen is not present, but if oxygen is present in the environment, then the anaerobe can undergo the process of cell respiration. Facultation means it can either be anaerobic or aerobic. So if oxygen is present, then the bacteria will become aerobic. If the oxygen is not present, then the bacteria will be anaerobic and it will undergo the process of fermentation. For aerotolerant anaerobes, these are anaerobic bacteria that are tolerant of oxygen. So you have some bacteria that they can be in the presence of oxygen, but they're not necessarily impacted by it, and these are kind of the opposite of obligate anaerobes, which means if oxygen is present, then the bacteria will die. So obligate anaerobes are not able to tolerate oxygen, but aerotolerant anaerobes can tolerate oxygen. They don't use the oxygen, but they can tolerate its presence. And then facultative anaerobes means that they can use oxygen in the process to undergo the process of cell respiration. There's also some organisms that rely on growth to be enhanced by the presence of carbon dioxide. And these particular organisms are known as capnophilic. So capnophilic organisms grow best whenever they have a higher concentration of carbon dioxide to enhance growth and development. Some nutritional requirements that are needed for bacteria include hydrogen, oxygen, carbon, sulfur, phosphorus, nitrogen. Those are some of the key elements that we've talked about being key essentials to life for many organisms. There's also special elements that bacteria require, such as calcium, iron, zinc, just like our own bodies. Organisms that are very particular about their nutritional requirements are referred to as fastidious, or also known as fussy. This means that if there aren't certain elements that are incorporated into the organism's diet, then they're not going to be able to survive. So the nutritional needs of organisms are also an attribute that we use to figure out how to classify them or to figure out how to identify what they are. The next attribute is that of biochemical and metabolic activities. This information basically is kind of taken from the use of different specialized medias known as differential medias. What we do to kind of evaluate the biochemical and metabolic processes of a bacterial organism, because we can't, dissect these bacteria. So what we do is we try to watch these reactions take place through experiments. So we can watch bacteria grow in a media environment. And in that environment, the bacteria will start to produce waste products or secrete materials such as enzymes. And then we can through a bit of manipulation, kind of use indicators to help us figure out if those particular enzymes, proteins, or products are being produced by the bacteria. And so one example is that of Staphylococci and Streptococci. These are two genres that express enzymes that are called hemolysins, which are used to break down red blood cells. And we're able to kind of understand the differences between Streptococci and Staphylococci because of this unique characteristic. So what happens is the Staphylococci will secrete a hemolysins when grown on an agar that has blood inside of it. And the hemolysins will cause a rupturing of all of the red blood cells surrounding the bacterial colonies. And so when you view the bacterial colonies after incubation period, you can see a clearing zone that surrounds the bacteria. And that clearing zone indicates that you had the bacteria secrete an exoenzyme known as the hemolysins, which are used to break apart red blood cells. And some Streptococcus bacteria do this as well. And because of that, we're able to use those particular enzymes that are being released, or hemolysins being released, to identify what bacteria are growing on a plate. Other bacteria can be identified through the production of different gases, such as carbon dioxide or hydrogen sulfide. If you ever try to grow salmonella in a laboratory, and you grow it in a media called triple sugar iron agar, you can see carbon dioxide being produced in the form of a bubble that gets starts to form inside of the agar, because gas is produced by the bacteria after a carbohydrate source is digested. So gas is released, it has nowhere to go, so it gets pushed to the bottom of the tube, and then it causes cracks to develop inside of the media. So if you see that in results, you know you have carbon dioxide production. For hydrogen sulfide production, we add, you can really tell the gas for this production, but sometimes you can add iron to the media, and the iron would cause the conversion of hydrogen sulfide into sulfate, and that provides a precipitate if the bacteria reduces the iron inside of the media. And so you can see this black precipitate form, and that'll be indicative of whether or not you have hydrogen sulfate production. But this hydrogen sulfide here is just gas, and sometimes you can really tell if it's hydrogen gas or carbon dioxide being produced. It kind of depends on what the organism is. in your knowledge of the respiration system. So back